interest. From where will you isolate a gene of interest? Alright, so you won't get it in the shop obviously. So you need to take a genome. Alright, whose genome will you take? You can take a human genome, you can take a plant genome, from wherever you want to isolate the gene of interest. So genome means the entire set of DNA is there. The complete DNA of the organism is there. This is the genome. Let's say this is a human genome, and from the human genome what I'm going to do is I'm going to isolate a gene of my interest. So gene of interest is GOI, and I have isolated the gene of interest. Okay. Now let's say this is human insulin. Just to make it interesting, I'll call it human insulin. So this is human insulin. Alright. Human insulin gene. From the human DNA I have isolated it. So my gene of interest is ready. Now what is the next step? Generation of recombinant DNA by insertion of the DNA fragment into a vector. So now what I need to do is I need to take this gene of interest and I'm going to need a vector. What is the meaning of a vector? In simple terms, we can call it a vehicle. A vehicle to carry the gene of interest. Alright. So I can take a plasmid. Let's say this is a plasmid. I've taken a simple plasmid. Just now we saw what is a plasmid. I can take a plasmid and I can insert this gene of interest into the plasmid. So you can see what I have done there. I have introduced the gene of interest into the plasmid. So now, because this is something I have created, what have I done? I have created a recombinant plasmid. Why is it a recombinant plasmid? Because I have taken a normal plasmid from a bacterial cell, I have taken a human insulin gene and I have inserted it. This is a new combination. Till now it was not there in the bacteria, neither in the human. So bacterial DNA plus human DNA, I have made a new combination. So this is a recombinant plasmid. This plasmid is now acting as a vehicle to carry my gene of interest. Where will it carry my gene of interest? We are going into the third step now. Transformation of our DNA into the host cell. So now that will be transformed into E. coli. I can put it into the E. coli. I'm sorry. So that now this E. coli is carrying the recombinant plasmid. The recombinant plasmid, I have put this plasmid with the gene of interest into the bacterial cell. Alright, now some cells may take it up and some cells may not take it up also. It's possible. Transformation is not 100% efficient. Some cells may take it up, some cells may not take it up. So finally, what are we going to do? We have to do a process of selection. Which are the cells we want? We want these cells or these cells? We want obviously these cells because they have our gene of interest. So when I select only the recombinant cells. I select only the recombinant cells with the gene of interest. Alright, and I allow them to multiply so that all the daughter cells will have the gene of interest. And whenever these cells divide, even their daughter cells will have the gene of interest. So these are the steps in gene cloning. Alright, the steps in gene cloning are very important basic procedure for gene cloning. So in the rest of this chapter, this is what we are going to learn about these steps in more detail, how to isolate the gene, how to put it into the vector, how to do the transformation process, how to do the selection process, all these things we will be learning in the rest of the chapter. Is that clear? Thank you.